Up to date, a lot of research on sickle cell disease has focused on what's done in the circulation, and little is known about what's happening during the process of erythropoiesis, which is the process of red cell formation. And so what we wanted to do is to investigate how these red cells are formed in the sickle cell context, and to really investigate what's happening in the bone marrow niche, because new therapies like gene therapy and also bone marrow transplantation aim to target the bone marrow niche to perform this therapy. So we wanted to know if the bone marrow niche is ready for this kind of therapy, and if having any defects in the bone marrow niche could impact the therapies that are given through this pathway. So this was a bit the way the project started. And then when we went further, we were able to observe that there were defects in erythropoiesis in sickle cell, and we showed for the first time that ineffective erythropoiesis is actually occurring in sickle cell disease, because up to date, most of the work has shown that it's not present, and it's mainly present in beta thalassemia and other pathologies. But we were able to clarify that it is ineffective erythropoiesis is occurring. It might not be at the same rate, but it is present, and it's something that we need to consider when studying the pathophysiology of the disease. So this study was conducted in two phases that were, I will say, parallel. First, we used human samples from patients. We isolated stem cells from patients and healthy donors. We focused on the severe genotype of sickle cell disease, which is the SS, but we are now investigating other genotypes because we do believe that each genotype is different. And what we did is we did in vitro assays, so we differentiated the stem cells in vitro, and we investigated whether the process of erythropoiesis is impacted using cellular techniques, but also molecular techniques. So we wanted to dig into the molecular mechanism behind the defects that we were observing. And so here we went and looked at transcription factors that are important in erythropoiesis, like GATA1. And also we used an animal model. As I said, this work was in parallel because the patient cells were able to show us really specifically what's happening at each stage of the differentiation. So these two approaches, the human cells and the bone marrow, were done in parallel because they are very complementary. So as the human cells using stem cells from patients allowed us to really investigate what's happening at each stage of the erythropoiesis, using the bone marrow allowed us to look at what's happening in the niche as a whole. And so we were able to see how these defects in erythropoiesis are impacting the architecture of the niche, looking both at bone marrow and the spleen in the sickle mouse model. Yeah, I think that this work allows us to shed the light more about the bone marrow niche, and that this is an important factor in the pathophysiology of sickle cell disease that should be considered when choosing treatment. For example, we know that bone marrow transplantation is working better in children than in adults, but we don't know why. And this work has shown that it might be due to having a very disrupted niche with high inflammation in the adults, while in the children, since the onset of the disease is not the same, we still have a better niche, and so they are more responsive to treatment. So I think that this work will allow us first to choose the time point at which treatments should be administered to patients, and also it's an opening eye that following what's happening in the niche should be part of the follow-up of therapies, including gene therapy and bone marrow transplantation.